All right, so what I'd like to do is a little bit of two things. One is to share with you some of our research, which is kind of the royal hour, but some of us that actually did these experiments are sitting in the room. And then also to kind of try to summarize the day's events and end this session with discussions with one another. And at that point, I'm just going to turn it over to you all to, that's Texan for you guys, to to initiate interactions with each other. I personally want to thank all the speakers. I think it's been really an inspiring event. I'm truly grateful for the diversity of speakers and what you've presented. And I know, at least for me, every speaker has inspired new thought about how to look at this disease. I'd also like to gather from you later by email some of your thoughts on what the following year's conference might look like and what themes you'd like to see continued. That would be very helpful to us. Okay, so what I'd like to do is start with a little bit of our work, which is not happening. Okay. It doesn't move.
It's a remote with this, so you have to kind of point it this direction instead.
Court, point to you. Yeah?
No, it's the other one. Is there another smaller?
Oh, no, that worked.
Oh, did it work? Okay, good.
I pointed at you. Okay. Okay. And actually, it brings, talking about our work kind of brings together a lot of things that have been mentioned in the talks today. And much of it kind of really goes back to a sabbatical. I went on sabbatical. I home back and forth, as I mentioned, to Montreal. And I spent a sabbatical there in the fall of 2006. And my goal had been to go there with newfound enthusiasm for looking at how tumor cells, as opposed to non-tumor cells, have very unique energy demands and energy strategies. And I got there, and one of my two hosts was this fellow named Jack Antel, and he had other ideas in mind for me for the summer. And actually, this talk is the conversion of both of those here 7 years later. Okay, so what I'd like to do is talk a little bit about the energy requirements of tumors and glioblastoma in specific, the potential of the effect of that energy strategy might have on DNA damage and DNA damage repair, and how all of that might influence the immune privilege that's provided to the glioma by virtue of the fact that it is not rejected by the immune system. And then what I hope to do is, end with, can we learn from all of the lessons that we've heard today how to come together and integratively collaborate with one another or with each other to come up with new ways to kill these tumor cells. And I think it will take those kind of collaborations. I'm often reminded of a definition that I heard a woman that was the director of the Joseph P. Kennedy Foundation describe for collaboration, which was not just talking about working together, not just sending reagents to one another, not just even doing the experiments together, but by the time you finish, being moved by the experiments that you've done to understand the new knowledge that generates. So my wish for this group is that we have that kind of productive collaboration in the end. Okay. All right. Well, yes.
The solid black one up there.
This one?
That's the one.
Oh, yes. All right. So what are any cell's fuel strategy choices? And no matter what the cell is, whether it's plant or mammal or whatever it is, there are limited choices for the fuel that's going to operate that cell and its functions. Okay. Whoop. Okay. It doesn't want to. Okay. Well, no.
We've been very consistent all day.
Nope. Okay. I'm pointing it back. Oh, yay. Look, something happened. That one?
These two arrows,
Yeah, that's what I did. Okay. So fuel metabolism, a normal cell, as we've all been taught on those biochemical and metabolic pathways, for 90% of the cells in our body, our primary fuel source is the carbons derived from glucose. And the rate at which we break down that glucose can vary from cell to cell, particularly in rapidly dividing cells. But in addition, there's other metabolic pathways that are initiated by growth factors or cell division, whereby glycolysis in the cytosol, meaning those steps breaking down glucose in the cytosol, occur at a rate that's a little bit faster. And then if for the most efficient energy generation, one would think, those at a certain step in the pathway, pyruvate, as part of the mitochondrial pathways, generates the energy to make the ATP that that cell needs to survive. It's supposed to be moving onwards, but perhaps it doesn't want to move onwards. Oh, good, it did. Okay, that's high-rate glycolysis. It's a slow road to high-rate glycolysis today. Okay, and eventually there's other choices. So one can use carbons derived from glucose in the mitochondria. One can use carbons derived from fatty acids, say, in the mitochondria. One can use carbons derived from amino acids. And if one is a tumor, and often other rapidly dividing cells, those amino acids include glutamine. Okay. No. Okay. Well, I didn't mean to go backwards. We're stuck. Okay, I'm going to change. I'm sorry, it won't go forward. No. I'm not sure how my challenge could affect this thing, but apparently it does. You know. Nope, doesn't want to go forward.
There's something weird with the power. No, keep using that.
Okay. Can you guys change it?
Can you run it from the back?
Yes, please. Oh, there you go. So we've talked about that. So it turns out that probably the most efficient apoptosis-inducing stimuli happen when cells are using glucose-derived carbons in the mitochondria. Rapidly dividing, proliferating cells are usually the ones that are more sensitive to apoptosis, and those, like Marty discussed, that have been stressed, either environmentally or with chemotherapeutic agents or with radiation or drugs or environmental inflammation, shift towards the use of autophagy, meaning eating one's own organelles as a source of fuel. And eating one's own organelles includes eating organelles rich in fatty acid. So what we began to do is try to understand what tumor cells are doing, glioblastoma in specific. Okay, next slide, please. Okay, great. And so what we did was use a cell line known as 263. So 263 is a human cell line that we obtained from Keith Black in California. And like several people have discussed, those cell lines, especially Dr. Del Mastro, those cell lines are recreating themselves all the time. But nonetheless, this was our model cell line. And I bring that up particularly to talk about the differences between growing those cells in tissue culture and what is happening in a growing tumor in the brain. Because what we began to learn is those things are really different. So we had a hypothesis that there would be high-rate glycolysis, that we might be able to slow down using the simple compound 2-deoxyglucose. And we hypothesized that a really seriously bad tumor has the plasticity to change substrates easily from glucose oxidation in the mitochondria to glutamic acid or glutamine or other amino acids to fatty acids. So we just decided what we would do is throw in both inhibitors, 2-deoxyglucose, with or without a drug that blocks fatty acid oxidation, known as Etomoxir. It's one of a family of things that one can use to inhibit a key enzyme in that process called carnitine palmitoyltransferases 1 and 2. There'll be a test on the enzymes just after this. At any rate, so Etomoxir had been used in clinical trials in people that had congestive heart failure, moderate success with some liver issues. But we thought if the hypothesis is correct, that glioblastoma, like other very difficult to treat tumors, have the plasticity to adjust their substrate in the mitochondria in aid of survival by using autophagy or not, we would expect to see an enhanced amount of percent cell death in this cell line, and that's exactly what we got. So This is percent death. We've used every form of cell death measurement ever. Hence, we are the Center for Cell Death and Differentiation, and hopefully soon to be partially comparative pathophysiology. At any rate, that includes tunnel staining and annexin 5 and pick one. We've used all of those, including trypan blue and flow cytometric analysis of various types. And what we found is it's really hard to kill GBM in culture once they get going. They are truly hard to grow, but once they start growing, they're resistant to everything. So we treated them with either Etomoxir alone. Does that do a whole lot? Not particularly, because they maintain the capacity of using glucose oxidation and high-rate glycolysis combined. If we put in 2-deoxyglucose, does that kill them? No, it actually makes them really happy. So they begin to growth the rest. They're not growing so fast, but they're not dead. If we combine the two, Eto and 2-deoxyglucose, in this cell line over a short amount of time, like 48 hours, we can kill a lot of the cells in a statistically significant way in culture. So there is no immune system in culture, so this has always been kind of bothersome to me, on top of which this cell line is a cell line growing in our incubator. On the other hand, we're looking at, this is at 48 hours, and this is at 144 hours, but also bear in mind, at that point in time, those cells are starving because they've been in that same culture medium that long. So they've probably gone multiple processes, including the end of it, without some additional nutrient. So I bring that up just to say this cell line at least does appear to be sensitive to changes in environmental nutrient source and, their mitochondrial substrates. So then we said to ourselves, well, okay, what happens if we use the drug that's out there currently, standard of care, Temozolomide, and we were very disappointed because we'd throw Temozolomide in there and they mostly did nothing. They just sat there pretty happy with this particular clown. So here was the percent death with no treatment and the percent death with 1 millimolar Temozolomide in the culture dish. And then we started doing some combinations, including a combination of putting in Etomoxir combined with Temozolomide. And you can see that we were increasing cell death a little bit. Or the Eto alone, we got a little bit, but not that much. It was only that when we combined the two that we started to see better efficacy with the Temozolomide. So my suggestion is, having had this in this drug has been in human beings, that we might want to consider those combinations. And if that's true, we also reasoned, would we see an effect in terms of MGMT and its ability to repair the damaged DNA that we were inducing with Temozolomide? Could we make it better? And if so, what was the effect of Eto on that? So we began to measure. There we go. I don't know what I did differently. Do you? Okay. All right. So we began to measure DNA damage. This is just pictures of what I've just shown you of how those cells look in culture. Oh, no, we need to DNA damage repair slide. Here it is, sorry. Okay, so we began to quantify DNA damage repairs measured by the number of abasic sites as the result of the Temozolomide, Etomoxir, or Temozolomide plus Etomoxir. And again, in culture, you can see that DNA damage repair was going on pretty well with nothing. We add Eto, which again becomes an oxidative stressor. It increases the amount of reactive oxygen in the cell. I'm not showing you the data, but it's measurable, quantifiable. Temozolomide, indeed, like it's touted to perform in these cells, increased the number of abasic sites that had been damaged. It was when we combined them that we got a truly significant rise in DNA damage. We also have measures of how rapidly the enzymes to repair the damage happen. But as a suggestion, this said to us in culture, perhaps it's time to converge metabolic disruption with DNA damage repair as targets. Okay. All right. So that's what we had done up to this point. No. There we go. Okay. So we're going to stop, everybody take a breath, change gears, and we're going to talk about the immune system for a little while. Yes, sir.
What day did you do the assay after the treatment of temozolomide?
48 hours. Yeah, I believe it was at 48 hours after treatment. But good question. Okay. All right, so we're going to take a breath. We're going to think about the immune system. And then we're going to come back to those cells in culture, because that is the bottom line of what I'm going to hopefully show you, provided the projector cooperates. So unlike Betsy, like Betsy, I'm an immunologist. I'm very interested in antigen processing and presentation. But we're interested in it from the perspective of B lymphocytes, the ones that make antibody. You know, the famous ones that you can get Rituxan and rituximab out of. But we think about B cells, you know, as being the prototypical cell that has an antigen receptor, recognizes antigen, gets help from a T cell, can also process and present antigens in the way that Betsy described, and display those antigens in the context of MHC class II, HLA, DR, DP, or DQ, and then talk to a T cell that will help it become a better antibody-producing cell. Come about the time that I went on my sabbatical, the notion that B cells were important for autoimmune diseases was beginning to emerge, especially in a disease like multiple sclerosis, where it had always been touted to be very much an autoreactive T cell-driven disease. However, people were using several drug companies had been using Rituxan and rituximab, which are B cell depleting antibodies. And in some cohort of patients saw, my friend Amit Bauer warned me not to say they improved. They had lengthier time to progression, lengthier time before progression of the disease became worse when B cells were depleted. And that started implicating B cells a little bit in diseases that were thought to be predominantly T cell-driven autoimmune diseases. In addition, all those other diseases that Betsy mentioned, including type 1 diabetes and rheumatoid arthritis and several other diseases, there were promising results coming out with removing those B cells. So we were very excited because it took me all the way back in my history as an immunologist. It was also important because we were trying to understand, you know, why that was a problem, why getting rid of B cells was a good thing. And at the same time, Dr. Antel was using a drug called FTY-720, which is fingolimod is its other name. It's an S1P antagonist. So for Dave's interest, that means that cells don't emigrate out of the lymph nodes so well, they kind of get stuck there. And we were looking at it from the aspect of how does fingolimod work? Because when Jack handed me the data from the patients of the group that were efficaciously treated, meaning longer to progression, had no B cells whatsoever in their peripheral blood samples. They were B cell-less. So the notion was that those B cells got trapped in the lymph node, and yet Jack had no evidence whatsoever that the lymph nodes were big or they were full of B cells or that everybody was trapped there. But there was also no evidence for apoptosis necessarily. Our take-home message was there's something really important about B cells driving disease. So working through what that might be and thinking about antigen processing and presentation, and also thinking about TLRs, those same ones that plants have and immune cells have, meaning receptors that respond to environmental products of pathogens, or alarmones responding to damage or danger or brain trauma injury, as the case may be. What happens to antigen processing and presentation in B cells that are activated both by those non-antigenic stimuli, a TLR, for example? And we predicted in Montreal that what would happen was antigen processing and presentation would result in a B cell, or the absence thereof, stimulation by TLR. There is no antigen to induce this process of H2M or HLA-DM replacing CLIP, and that the consequence might be MHC class II ectopically bearing this invariant peptide. And so we began to wonder, what does that have to do with the price of eggs? But I'll try to explain our model. All right, so in the meantime, as Betsy described, we all have MHC class 1 and class 2 genes. Mine are different than yours. They're highly polymorphic unless you're my twin. And of those, the ones that we think about, which are on the B cells, are these MHC class 2 molecules. And so those are, again, highly polymorphic. So the grip, so to speak, on CLIP between my MHC class 2 and yours is a function of what amino acids make up our particular MHC allele. So we said, okay, what difference might that make in those genetic susceptibility and resistance cases and correlations to disease? So this is all of immunology. Betsy's done it much better than me going over this, but here's our B cell showing antigen to a T cell, and here's our T cell helping that B cell, or a different group probably helping the CD8. The difference was this one, where we've got TLR activation instead of antigen receptor. And while I was on that sabbatical, we discovered that indeed TLR activation results in CLIP expression on those MHC class 2s in the B cell in the absence of any antigen. So further work, and well, maybe further work. Can we? Oh, thank you. Okay, so the other thing that we learned is when you have a B cell and there's been no TLR activation and no antigen stimulation, and any cell that can come along and bind to MHC class 2 on that B cell in cells that are your resting cells, and now actually we know which ones they are, that are sensitive to class 2 mediated cell death. So we began to wonder what happens if this has CLIP in the groove and there's been an activation event. Does CLIP in the groove of class 2 prevent the cell death of what normally would be a negative regulator, getting rid of a B cell that potentially could be autoreactive? So we asked that question by maybe, can I have the next slide? Thank you. By looking at the impact of anti-class 2 antibodies as a surrogate for that T cell that could kill off B cells. I'll tell you about MKR4 in just a second, on resting cells, so that's this bar, versus those activated with CpG. So it's a TLR mimetic. And you can see that these are more sensitive to class 2 induced cell death than the CpG treated cells, unless I purposely put in a peptide, cleverly called MKR4 here. It has so many names, but it's the same peptide, that has a higher binding constant than CLIP for the groove of every known MHC allele as computed by my son Evan using an algorithm that he wrote to predict a peptide that we had synthetically produced. And you can see that if you add that peptide and now you come back with class 2 as a surrogate and you remove CLIP from the groove, that class 2 engagement results in cell death of that CLIP-bearing TLR-activated cell. All right. If we do this in an animal that has no invariant chain knockouts, the same is not true. If there's no CLIP, all cell death looks the same. And in fact, on the activated cells, everything kills those B cells. So what does this have to do with glioma? Well, possibly nothing, but at some point I argue not, that this might be quite important. So what we've learned is that stressful events, TLR activation, inflammation, danger, damage, lots of cell death, can produce signals that can activate the increased expression. This is supposed to be, can we hit any key? Yep, thank you. To put CLIP, as I've drawn here in the form of a camouflage flag, on a cell that can protect it from T-cell mediated death, right? We've recently learned that one of the signals that can put CLIP on cells is probably through what's called the P2X7 receptor when it binds ATP in its environment. So if you have a boatload of cell death and lots of release of ATP, in nearest neighbors that need to live, using benzoyl ATP, we can show that that also increases CLIP on other cells, not just B cells. So it's kind of a protective strategy for cells that need to live under conditions of inflammation or under conditions of lots of cell death. So what we think is that CLIP provides kind of an external camouflage that is part of that protective boundary of tumors, say, that include myeloid suppressor cells and Tregs, and the inability of that cell to die by T cell receptor engagement of MHC. And so we went on to kind of look for that. Can I have the next slide, please? Oh yeah, there goes inflammatory cells. We also predicted that those would be quite pro-inflammatory, which, Betsy, kind of fits a little bit with one of the descriptions that you gave between the difficulties of the mutant of associating DM with DO. One would predict that the ones that never got DM would express more ectopic CLIP, and that might contribute to the inflammation from lack of peptide exchange. All right, so next slide, please. Okay, so this is my last science slide. I'm so proud of this one. This was done by, so the first thing I have to tell you is that we tried everything with 263s and 261s and every other kind of glioblastoma cell line to see if we could see MHC class 1, MHC class 2, FAS, any death-inducing receptor, and they were always dead negative. And we looked for perhaps the expression of CLIP, thinking that would be part of their protective strategy. Saw nothing. And then it finally dawned on us, which is totally obvious, that growing in a tissue culture dish is really different than the microenvironment in the brain where this tumor is growing out of hand. And there might be a lot of other influences around that brain tumor. So thankfully to Dr. Soren Singal as the neurosurgeon and Dr. Eka Kobi-Fonkum as the neuro-oncologist, we managed to get an IRB in place to receive immediately ex vivo explants from glioblastoma, where Dr. Sanjeev Mukherjee started staining them. And lo and behold, so far, all of the cells that he has stained and done immunohistochemistry illustrate that the tumors coming right out of the brain of the person express ectopic CLIP. So that says to us they've got another barrier. Not only do they have a barrier against drugs and radiation and immunogenicity, which is probably the wrong T cells around them anyway, but they have this camouflage flag. The good news is it predicts that a high affinity, really small peptide, like the one computed to compete with CLIP, might be very useful in terms of treatment for GBM. So I'm working with a company, in full disclosure, I work with a company called VG Life Sciences, and the peptide was synthesized in 2007 or 8. And we now can send it to anybody that wants to try it. I mean, we'd love to have you try it. It's potently anti-inflammatory. And so we'd be more than happy to share that. But the peptide itself is also in preclinical studies at a contract research organization to obtain safety and pharmacology for its use in animals, including mice, rabbits, and macaques. So that's where this study lives now. So with that, I'm going to stop and change gears, take a breath, answer any questions that you might have, and then try to facilitate our next discussions with each other before dinner. Questions? Hi, Betsy. Sure.
So my question is, are you saying that you're seeing is the F that you're actually seeing class 2 on the glioblastoma explant? And then G is CLIP, but I'm looking at F first for the red.
Okay, so the red is MHC class 2.
Right, and
which
I'm
you can
asking whether, so F, isn't that F
It's-
up in the top right?
Help me, Sanjeev. Okay.
We
So
saw-
this is DAPI.
So this is not the best picture.
Okay. Okay. I hit him up for this like yesterday.
So I got it from my.
This is DAPI.
Yeah.
DAPI?
This is DAPI, but we did get Cellular.
This
3
one.
patients out of 4. And here you don't see, the signal is not that great, but we did
The green is clip,
Class
the red is class 2. It's poorly visible, but you can see it here.
This thing is not.
But is there a
I'm
dot
just
behind
trying to
those?
figure out I was just trying to figure out what the result was. So,
Okay,
but-
sorry.
So that's okay. So the idea, so my question is whether you thought you were seeing clip staining
In
on
class-
the tumor itself that was somehow separate from seeing class
That-
2 on that tumor.
To the best of our knowledge, I mean, based on morphology alone, this is really, really new. It doesn't look to be other cells that are bearing clip. It looks to be the GBM, but this requires a lot more study. In vitro, when we looked in vitro in tumors, we never see it. I take that back. We never see it unless we treat them with BZ-ATP, and then they become clip positive in vitro. Yeah.
Yeah, so I'm just still trying to understand whether you think there may be other surface receptors or surface molecules that can bear the clip besides MHC class 2. That's what I'm really trying to figure out. Because
It's-
I'm looking at your class 2 staining and what I think is
Could be
red.
class 1, maybe, by cross-presentation, would be my only other guess. I don't know of other cells that could, bear clip in this way, but that's not I have no idea. The only other candidate would be, we have seen, and so has Nulamuni, cross-presentation of clip through class 1. So it could be class 1 bearing less class 2. And again, very early data, but my point for this is two. One, that we need to think about the difference between in vitro and in vivo in human tumors. And two, some of the things we think we can't see in vitro are there in vivo, and they may be helpful in terms of how we approach treatment. So-
This is the proteolytic process?
Yes. And we've ruled out intact CD74 in most of our studies where we're looking at clip on other tumors like B-cell tumors. You can see it no problem. Just glioma, never. Not unless you look from in vivo tissue. Okay? And the B-cell tumors are like Dowdy and Raji, and they all have clip that you can detect with the antibody, and it's not the same as CD74 staining. So we're pretty sure it's the proteolytic product. Okay. 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 All right, so I think what we should do is just open the floor up for discussion amongst everybody. And I again want to thank everybody. I think this has been really stimulating, and for me, a really special event. Thank you for participating, and I'll turn the floor over to whoever wants to talk.